Well, good evening. I'm Susan L., Vice President, Executive Director of the Foundation for Barnes-Jewish Hospital. My pleasure to welcome you. We are so excited that you could join us for this really stimulating leading-edge medicine program this evening. The presentation will give us an inside look into the next generation of treatments in orthopedic medicine. Now, before we begin our program, I would like to take a moment to thank you so very much for your generous gifts to the Foundation for Barnes-Jewish Hospital. Our mission of enriching lives, saving lives, and transforming healthcare would not be possible without your partnership. Together, we are advancing breakthroughs in medicine, preparing a brand new generation of rewarding careers in healthcare, and making a difference, a true difference in the care of our patients and the families we are so privileged to serve and care for. Healthier lives and healthier communities. We are grateful that you are with us on this journey. Now, at this time, I'd also like to welcome our Foundation board members who are with us tonight and Foundation team members. You will be able to tell who they are because we're all wearing this silver badge. So if you've not had the opportunity to say hello and meet, please do so. And Dick Miles, who is chairman of our board, will be coming up at the end of the program to conclude and make some final remarks. So let's get on with the program. Our guests tonight are Dr. Regis O'Keefe, the Fred C. Reynolds Professor and Chair of the Department of Orthopedic Surgery at Washington University, and Dr. Farshid Guilak, Professor in the Department of Orthopedic Surgery at Washington University and Director of Research for the Shriners Hospital for Children in St. Louis. Dr. O'Keefe is a great partner to the Foundation, and he will lead off the program and take us into the featured presentation. Dr. Guilak, the co-director of Washington University's Center of Regenerative Medicine, will share with us just how his laboratory's research in regenerative medicine accomplishes remarkable innovation that allows us to grow our own custom cartilage to repair our joints. And I've heard from many of you that, I wish I had heard this maybe 5 years ago, because I've had this replaced, and the science of the regeneration and joint replacement is just moving along really quickly. Arthritis and joint diseases are extremely painful and very debilitating for so many of us, and remarkably, it affects almost 1/4 of all working adults. Can you imagine having your own personal cells grown in the lab to repair your joints, rather than having to have your hips, knees, and other joints replaced? Please join me in welcoming Dr. Regis O'Keefe, Chair of the Department of Orthopedic Surgery at Washington University. Dr. O'Keefe.
Well, thank you very much, Susan, and I want to thank the board and the foundation for the opportunity to be here tonight. It's such a great place to be. I have a wonderful view out the back, so and I appreciate your attention. So I get to talk about my favorite thing, orthopedics. So I'm really excited to be here, and I hope I make you excited as well about what I'm going to tell you about, and also the kind of information and the innovation that you're going to hear from Dr. Guilak. So many of you are in business, and we're in the business of care. We're a caring organization in the Department of Orthopedics, and when we think about what our mission is, it's about being the best. It's about achievement, and we build that with people and values and culture, like many of you have built your businesses and have thought through that process. And we want to be the best in areas like clinical care, education, research, and leadership. And we put this thing all together in our department. And what I want to tell you is it's an amazing success story. So I want to give you a little background on the department that Dr. Guilak and I work on. And this shows the development of our faculty and staff. And you can see since 2010, we've almost doubled in size. And we have 569 people who now work on a daily basis to take care of patients in this community. These are the 66 clinical faculty members that I have, along with 13 basic science faculty that really make us special and a national and international leader. Now, one of the things that I get to do as the chair of a department is recruit people. And I've recruited 31 people to the Department of Orthopedics. That's a lot of dinners. I've managed to exercise and keep that going, which is a really good idea. But these are really amazing people, and these are leaders in the field. There's already leaders. Dr. Guilak is one of these people. He came from Duke University, where he was a distinguished professor. But there's many other leaders here. And what I'm really proud of is that many of our young faculty working in an environment with people like Dr. Guilak and Dr. Kalhisi, who's a total joint arthroplasty surgeon, Dr. Barrick, another total joint arthroplasty surgeon, have the opportunity to develop as national leaders. Now, we really do think about leadership, and we have a remarkable history of leaders that are heads of national organizations. So we're not only doing things locally in terms of our impact is nationally through organizations. And I want to point out this is Dr. Guilak as president of the Orthopedic Research Society. I was also the president of the Orthopedic Research Society, and one of our other colleagues, Dr. Linda Sandell, was. I think we're the only university that I know of that have 2, let alone 3 presidents of the Orthopedic Research Society. So we're very proud of that accomplishment. Now, it really begins a lot with clinical care. That's the platform where we can do things like education and launch research programs and have innovation. And just a little bit about our clinical care, and I mentioned again the size and growth of the department. This shows the outpatient office visits, and you can see that when I came in 2014, we had about 118,000 patient visits, and this year we had about 165,000 patient visits. So we've had this really extraordinary growth. And again, that points out to the fact that I think we're delivering care that is meaningful to this community, and that's driving success. It's driving the opportunity to recruit those 31 people who are increasingly going to have an important difference in the health of this community. So that's what we're really all about. And then this is the surgical case increase that we've had as well. And we do about 15,000 surgical cases on a yearly basis. Now, I think one of the things that I want to point out, and I think Susan mentioned this, that about a quarter of people have orthopedic diseases. And when I went into orthopedics, I should tell you, I guess I can tell you the year. I decided on orthopedics in 1984. I was at Harvard Medical School, and it was a very small specialty. And the transformation has been unbelievable. And this just shows the cost of care. This is from the Kaiser Family Foundation here, this data. And what you can see is that musculoskeletal diseases are the 3rd most costly condition in the United States. They're more costly than our neurologic diseases, more costly than cancer. And why is that? Because so many of us get those diseases, and particularly joint arthroplasty. Joint arthroplasty is the single most costly condition in the United States. So again, we have an amazing impact on this community by the types of things that we do. And how do we do it? Well, I want to just show you that when you take a look at something like total joint arthroplasty, this is 2019 right here. This is the projected increase in knee arthroplasty in the next 2 decades. This is hip arthroplasty. So increasingly, we're going to see more and more of these cases. And if you look over the next 10 years, and this is by SG2 Health, which is a national consulting company that Barnes-Jewish Hospital and many other leading organizations uses, they're predicting that there's going to be a 78% increase in total joint arthroplasty in the next 10 years. There's going to be 134% increase in shoulder arthroplasty. So there's going to be increasing utilization. And why is that? Because we're able to do it in older people, we're able to do it more successfully in younger people, and our population is aging. So these are, again, very important procedures. And I just want to point out this patient I saw last Friday. This was a 67-year-old gentleman with a really severe neurodegenerative disorder. He had been in a wheelchair for almost a year, unable to walk because associated with his neurologic disease. He had this really bad hip that you can see here. And I operated on him this past summer, and I saw him in the office on Friday, and he's now 6 weeks after surgery. He has no pain, and he's walking. This has made a difference. He was getting in a situation where, again, our independence, particularly as we get older, we want to be independent. We've been so independent. We've been successful our whole lives. There's nothing better than independence. It gave it back to him. So these are amazing procedures that we're able to do. So how do we do it at Barnes? And I want to share something I'm really proud of. We do an amazing job of this. So Vizient gets data from all the major medical centers in the United States, Harvard, Johns Hopkins, the Mayo Clinic, UCLA, Duke University. You can name all those places. And what I'm showing you here is if you take a look at our volume and total hip arthroplasty at Barnes-Jewish Hospital and West Conner, which are 2 hospitals, you add them together, we're number 4 in the United States in volume. So that's really something to be proud of. And one of the things that's sometimes seen is, the more you do of something, again, in a business, the more you do of something, the better you should do it. Well, how do we do? Well, one way that you measure how successful you are is how fast do patients get out over surgery? How soon are they walking well? How soon are they able to go up and down stairs? How quickly can they go home? And what you can see is we are number 1 in the United States. We have the lowest length of stay in the United States. I just show that Rush University, NYU, a place called Mayo Clinic, and a place called Johns Hopkins aren't doing as well as we are. So again, I'm really proud of the faculty at Washington University and the kind of performance they have. So we do a really good job with osteoarthritis, and I hope you agree it's common, it's costly. But I'll tell you that we got a problem. I love doing surgery, and I always tell patients when I come in, particularly when they don't when they don't have a problem, they come in, they say, I got this problem. I always say to them when I don't have to operate, I say, I'm a surgeon. I love to operate. I would love to operate on you, but I'm telling you, you don't need an operation. And I would rather be in that position where even those challenging patients, that patient I told you about, that 67-year-old gentleman with a neurodegenerative disease, he had to have a big operation. It'd be better if he didn't. And we're kind of right now at this stage, we have no medical treatment to delay progress. We have no regenerative treatment to restore joint tissues. And this is where Dr. Guilak comes in. He's going to really define for you what's the future, what's possible out there, where can we go in the future. And I just mentioned he's really one of the leaders in the United States in research, musculoskeletal research, and around the world. He's a professor of orthopedics, developmental biology, and biomedical engineering. He's also the co-director of the Washington University Center for Regenerative Medicine. He's a past president of the Orthopedic Research Society. He was the chair of one of the NIH study sections that decides who gets grant funding, the skeletal biology, structure, and regeneration study section. And he has more than 300 publications, numerous patents, and he's a recipient of numerous national and international awards. Now, I just do want to share a couple of personal things about Farsh. He also is a father. He has a child, a daughter, who just graduated from Washington University. His son, Justin, just started at Rice University. Took a 2-week trip to the Galapagos, and he was off the map. I couldn't talk to Farsh for 2 weeks, so made sure I talked to him when he left and before he come back. The other thing that's interesting, I'll tell you, I had recruited Farsh, and he came up for his recruitment visit, and I didn't know that Farsh was a racquetball player. And I don't know if people, that's a really challenging game. It takes a lot of energy. And there was he came up, and there was a racquetball tournament, and it was a professional tournament. And I found this out later that he said, well, I'm going to play some racquetball here. He entered this professional tournament. He turns out he's been in the top 100 in the world in racquetball. And so Farsh came, and I came in on Monday, and I thought, I wonder how the rest of his trip went. When I hear, he won the tournament, I said, what tournament? The professional tournament. He won the tournament. So Farsh came up, and he had that first great experience of winning the tournament. He decided to come here, and I'll tell you, we've been winning ever since. We at Washington University, and I think this community, we've been winning ever since. I think you'll agree with me. So, Farsh, thank you so much again for being here this evening. I look forward to your presentation.
Thank you so much, Dr. O'Keefe. So I moved here a little over 3 years ago, and you're looking at the reason that I moved here. So Dr. O'Keefe is widely recognized as the top orthopedic chair in the country and probably in the world because he's excelled at all of the different areas in orthopedics, both at leadership, research, and clinical care. He's practicing always in surgery, but running a nationally funded research laboratory. So racquetball was the second reason that I came here. For those of you who don't know, St. Louis was the home of professional racquetball in the 70s and 80s, and I've been very fortunate. My doubles partner now is the 7-time world #1 player, Marty Hogan, who grew up here and plays at the JCC with me. So it's been really fun being here. So, as you heard, I'm going to talk about arthritis a bit, and I like to ask, but I know the answer, who here has had joint pain, arthritis, or knows someone who does? And everyone raises their hands because it's so common. And what we're trying to do in the laboratory is come up with alternatives. And I'll tell you why we need alternatives, because it is a great surgery. And for certain patients, as Dr. O'Keefe described, there's nothing better. It's one of the highest satisfaction surgeries that's available out there. But certain patients, it would be great if they could avoid it. And so as humans, we are exceptionally evolved. And as we evolved, we unfortunately gave up some abilities that lower animals have. And one of those abilities is the ability to regenerate. So this is the limb of a salamander or a newt. And you can see if it's cut off, it completely regenerates. So it forms what's called a blastema. It's like a little bud of cells. And those cells regrow every other cell type that was in that limb. And of course, we have none of those abilities. The only thing we can regenerate in that capacity is the tip of your finger, where you can actually regrow some of the skin and a few other things. And then inside our body, we can regenerate bone to a certain extent. So if you have a fracture, in about 6 weeks, it'll heal. And in a good case, it'll completely heal and it'll regenerate. Some of our other tissues, muscle, liver, have some regenerative potential, but our joints don't. And so what we're really interested in is the cartilage in our joints. And this is a knee joint. This white tissue is what we're talking about, is the cartilage, the articular cartilage. And what happens if it gets injured or degenerated, if you look at this is a broken piece of cartilage. It's like a pothole in the road. It's just going to keep growing and get worse and worse. And there are currently no ways to effectively repair this. And what you can see, something I like to point out, there's no red here. There's no blood, because cartilage has no blood vessels in it. And this is because of its unique ability to withstand 5-10 times your loading, your body weight in loading. It doesn't have blood vessels in it, so it never heals. So something like this starts off painful, but then degenerates into your entire joint, and you need a joint replacement. And other types of arthritis, like rheumatoid arthritis, which is very different. It's an autoimmune disease. It's very hard to replace every joint in your hand or in your entire body if you have a disease like rheumatoid or psoriatic arthritis, which affects everything in your body. It even affects your mind. It causes depression, fatigue. It's a systemic disease. So what are the treatments? Well, the first thing I say is the best thing you can do is weight loss and exercise. But beyond that, you can take Motrin or Celebrex, and then what people do is take these nutraceuticals like glucosamine and hyaluronic acid. Unfortunately, as we're doing more and more studies on these, they really have no benefit. So there's a great placebo effect with these, but in terms of preventing arthritis or treating the disease, there is no benefit at all. So we're basically saying, go from taking Motrin to having your joint replaced. And we have nothing in between to prevent that joint replacement. And as you saw from the pictures Dr. O'Keefe showed, we have to cut away large parts of the bone and insert these metal prostheses with plastic on them. And it's a great surgery. If you're in your 70s, it's going to outlast you. But this is a patient that Dr. Clohissy sent me the x-rays on. And you can see this patient has already had one joint replacement. So you can see this long metal stem, the bone's been cut off here, this cup has been placed here. The other joint looks okay on x-ray, but when he went in there with a little scope to look at it, you can see the cartilage is completely gone. This is all bone that's exposed. This patient is 22 years old, had a joint replacement at 18, and needs a second joint replacement at 22. And this is the kind of patient we want to avoid doing joint replacements on, because these things only last 15 years, maybe 20 years. So if you're 22, then you're going to have another one at 42, and then it gets worse after that, probably one at 50 and then 60, and each one has a higher risk for infection. So if we can put off that joint replacement, we would be much better off for these patients, and the number of young patients is dramatically increasing for reasons like hip dysplasia, but also obesity and increased activity even. So this need for tissue regeneration overall is really growing. So it's not just our joints. Joints are probably number one, as you saw from Dr. O'Keefe's talk, but if we look at all the parts of our body that are failing with time and with age, it's costing the economy about $1 trillion a year. And that's joint replacements, skin failure like diabetic ulcers or burns, heart surgeries, organ transplants. Here you can see the number of transplanted organs that are available, and the patients on the waiting list just keep going up and up, and the number of organs aren't increasing. So there's a real shortage of transplant organs, hearing loss, vision loss, dental reconstruction. So these are areas that our center at Washington University tries to deal with in regenerative medicine. I'll tell you a little bit about what regenerative medicine is. So when I started in this field, my background is in bioengineering. So I'm trained as an engineer, but with biology also. So we study how we can use engineering principles, so design and predictive modeling and rebuilding things to the human body. And when I started in 1988 or so, this was the cover of Life magazine, and it was about how do you rebuild your body. And you can see it's all metal and plastic. So here's a joint replacement, here's a prosthesis, the artificial heart, some catheters, and so on. And about 10 years after that, this field started that I was very lucky to be involved in at the very beginning in the early 80s and mid-80s, which was called tissue engineering or regenerative medicine, where we thought, well, we don't always need metal and plastic. Can we regrow body parts? Can we take cells, living cells, and get them to regrow all of the parts of your body from muscle, bone, organs, cartilage, skin, blood vessels, everything else that we need? And the way that this can work, the concept of regenerative medicine, is that we harness living cells to recreate your body. Instead of using metal and plastic, we take cells from somewhere, and I'll tell you where we can get all different types of cells from your body or from donors, and use them to regrow what's missing in your body. And so one of the main ways that we do that is to use what are called stem cells or cell therapy, where we inject cells into the body, and they can hopefully help treat disease and regrow tissues. The main area we work in is tissue engineering, and it's really what the name says. We're engineering living tissues, and that's done by combining living cells with materials that the cells can grow on, and then giving them all the right signals so they know what tissue and what organ to grow in the right shape. And then another area that we're working in, and I'll only show one or two slides on this, is gene therapy, where we can introduce genes and gene corrections into the cells in your body or into cells on the outside and then put them in your body. And this can be used to correct genetic defects, or the way we're using it is to deliver drugs for a long-term basis by inserting those genes into cells in your body. So what are cells? Going back to high school biology for everybody, they're really the fundamental unit of life. It's what we define as life, is something that's living and has DNA. And the DNA is the code that tells a cell what to do. And cells read off this code. So the genetics is the sequence of this code, these 4 nucleotides or base pairs that define the sequence of the DNA. And then the other area that defines what a cell does is called epigenetics. And this is actually Dr. OKeefe's area of research, which is basically, to describe it simply, it's how that DNA is folded and exposed. So it might have the same sequence, but how the cell reads off that sequence and which areas are read is what tells a cell to become a nerve cell versus a fat cell versus a blood cell or a muscle cell or a cartilage cell. So all the cells in your body have the same DNA sequence, but the way that DNA is folded is what tells them what type of cell to be in your body. And if we want to grow a joint, we need to have all different types of cells, right? We need not just cartilage, but we need bone, we need tendon, we need ligament, we need fat, we need muscle, we need blood and blood vessels, other connective tissues. So how do we get all these different cell types from one cell? This is where we turn to stem cells. And your body, turns out, is full of stem cells. And stem cells have a very simple scientific definition. It's a cell that hasn't decided what it wants to be yet. Any of you who have a teenager, think of a stem cell as a teenage cell. So you want it to grow up and decide what to do. The other thing that a stem cell can do is replicate, and that's what you don't want your teenager to do. So this is the very simple scientific definition of a stem cell. It's a cell that can create two of itself, or it can turn into what we call a differentiated or specialized cell type. And so from a single stem cell, we can get all different types. And there are different types of stem cells in your body, those that form musculoskeletal tissues, those that form blood cells, and those that form other organs, and so on. So it turns out we have stem cells all over our body. The place that they were originally discovered was in the bone marrow. So all of our blood turns over very rapidly. So every second, we make a few million new blood cells because they get worn out as they travel around your body. And if we didn't have stem cells in our bone marrow, we would be out of blood cells in a couple of months. And the place that they turn over the most quickly actually is in your gut. Every 2 to 3 days, the lining of your gut turns over, and stem cells in your gut make a new lining. So they have to divide, make one stem cell and then one gut cell. And this keeps happening. Your skin turns over every few weeks. other organs also turn over this way. So can stem cells cure everything? Well, I'm sure you've seen ads like this. It says, what are stem cells? Stem cells are specialized cells that know what you need, where you need it, and they go there and they become it. And you've probably seen these in in-flight magazines and on billboards. Unfortunately, this is not true at all. All of these clinics that are offering stem cell therapies are actually not approved by the FDA. Most of them are not even dealing with stem cells. They're taking what we call scientifically gunk from somewhere in your body, like fat or bone marrow, and just injecting it into your joints or into your bloodstream. And there have been a number of cases of really bad side effects and adverse events from these types of procedures. People have had tumors forming from stem cells being injected into their spine. Last year, a whole bunch of people, 4 or 5 people, were blinded by stem cells being injected into their eyes, both eyes at the same time, and countless infections. So we don't recommend any of those stem cell therapies that are being offered out there now because they haven't been approved by the FDA and they haven't even been shown to be effective. Again, it's this placebo effect because you're going in there and you're paying cash, $5,000, $10,000, and most people feel a little better after they do that, but it doesn't last and it doesn't have an effect. And these clinics have really proliferated. At last count, it's close to 600 sites all over the country that you'll see advertising these stem cell therapies. So if just injecting these cells, whatever they are, doesn't work, what can we do to make them work? And so what we do is reprogram them and try to get them to regrow organs and parts of your body outside the body in a lab. And the way we do that is we take those stem cells from somewhere in the body, and I'll show you where. We reprogram them, so we recode their DNA, and I'll show you how we do that. So we get them to make the tissue that we want them to. We put them on a biomaterial scaffold, or a material that gives them a template. So once these cells are taken out of your body, they don't know what to make. We tell them what shape to make by giving them a scaffold material, sort of like a building scaffold that's the exact shape they need to be. They grow on it, and as that scaffold melts away, they create new tissues and replace it, and we've told them what tissue to make by giving them vitamins and hormones that tell them to become cartilage or bone instead of nerve or muscle or blood. And then once we've grown them, we implant those in the body, and that's where we need surgeons because we still need to get these back into the body somehow. They're just not metal and plastic, they're alive. And so where do we get stem cells? I told you about bone marrow. It's a very rich source of stem cells, and there's stem cells there that can make all of your blood cells, red blood cells and white blood cells, and there's other stem cells in there that can form various tissues of your body called mesenchymal stem cells, and they can make all of the musculoskeletal cells. And it turns out these mesenchymal stem cells, in one form or another, are in multiple tissues of the body. They're in muscle, bone, cartilage, placenta, various organs, and we've just more and more discovered how to isolate them and how to find them. And more recently, a discovery by Dr. Yamanaka in Japan got the Nobel Prize in 2007 because he found that we could take any cell from the body, give it just 4 defined factors, and we could turn it into a completely pluripotent stem cell. Pluripotent meaning that it can make any tissue in the body. So we can take a skin cell, or even at the Shriners Hospital, we take urine samples from kids, and the cells that just come off their bladder into the urine, we can reprogram and turn into what's equivalent of an embryonic stem cell without touching an embryo. We can just take a piece of skin and make these cells that'll make any tissue in the body. So there's no shortage of cells where we can get this. And the place that we have large numbers of cells that we've gone to over and over, and we actually discovered this a little bit by accident, is in subcutaneous fat. So we all have lots of fat, and we need to restore that fat. So there are stem cells in your fat that whenever you need fat, they'll make more fat cells. And unfortunately, when we eat too much, we make a lot of fat cells, but we don't necessarily need them. But we can take just 50 grams of fat using liposuction or a similar technique, and from 50 grams of fat, we can get 1 billion stem cells in 2 weeks. And so there's all these different places, bone marrow, fat, muscle, where we can just pull out cells very easily and use your own cells now to regrow these organs. So how do we create a template? There are all different ways to do it. So if you want to grow something hard and solid like bone, we can use a 3D printer and print these different scaffolds in the shape of a hip, for example, or in the shape of a jaw. These are biomaterials that have mineral in them. We can create them with a 3D printer in the exact shape they need to be. And as I mentioned, they slowly melt away while the cells go into all these little pores, all these holes, and they create living tissue. So over time, as this synthetic material disappears, your cells make a brand new material. So cartilage, unfortunately, isn't hard and solid like bone. It's actually soft and absorbs shock, and it's very flexible. So when we try to make cartilage with a 3D printer like this, it comes out hard and solid. So it didn't work for us. After years of experimenting, we developed a technique where we can make something really flexible and tough, something that's strong but can be compressible. And we did that using not 3D printing, but a technique we call 3D weaving. So weaving, as you know, if anybody's done any weaving, we take fibers and pass them over and under each other to form a fabric. And what we did was make this fabric out of biomaterials that are compatible with the body. So if we look at this, we have now 600 of these little fibers. And think of these fibers, if you ever had stitches that melt away, they're called resorbable sutures. They're made out of polyesters like polyglycolic acid or polylactic acid. They slowly melt away. They leave a little bit of residue, but then they disappear. Your body just turns them into lactic acid and they disappear. This is a polymer called polycaprolactone. It's a kind of suture that takes a long time to melt away. We took 600 of these and wove it into a three-dimensional fabric. And so you can see this is a weaving machine, a loom that we built in our laboratory, and you can see the fibers that are going in three different directions, the X, the Y, and the Z direction. And what we end up with is a fabric that's biocompatible and it's porous. So there are holes here, and you can see we fill it with cells. Each little green dot here is a live cell that we got from liposuction and digest it out. And you can see the fabric, we can make it thin or thick. We can weave it tightly or loosely, and after a year and a half of weaving different combinations, we came up with a combination that matched the properties of your cartilage exactly. And this way we could put cells on it, and it acts like cartilage right away, and over a year, the cells create new cartilage, and this material slowly melts away. And this image was so pretty that the Museum of Modern Art in New York actually had a display of science and art, and we were able to display there also. So now that we have this, sorry, it went a little fast. So if we want to make now a joint in the exact shape of your hip, we can take an image of your hip using three-dimensional imaging, like an MRI or a CT. We create a mold in the shape of your hip, and then we weave a construct in this fabric that has properties of cartilage in the exact shape of your hip. And you can see the pores here, and this is a 3D image of that. You can see as we slice through this image, you can see all of the fibers and all of the pores where we put cells. This is the actual construct in the shape of a human hip, and then we take fat stem cells, grow them on there for 4 weeks, and we've actually grown a living hip outside the body. And so this acts like a hip, and it has cartilage properties like a hip. And we're now testing this in animals that actually have hip problems, and we're finding that immediately after they've had the surgery and replaced their hip, they're out of pain, and 6 months later, this artificial cartilage has lasted and has worked really well. Now, the FDA wants us to repeat this study for 1 year in an animal, and if that works, then we can actually start testing this in people who need hip replacements. So we're well on our way with that. So the concept would be, if you have osteoarthritis, think of that 22-year-old patient who doesn't want a joint replacement and just wants to put off that joint replacement for a few years, if at least, maybe longer. We can get stem cells from anywhere, probably from fat or from bone marrow. We grow them up. We create a hip scaffold in the shape of her hip, grow this, and then instead of cutting away all of that bone, we just resurface the hip cartilage that's been damaged, and hopefully put off that joint replacement for 5-10 years, maybe even be able to repeat this if we can. So we're very excited about this. As I mentioned, it's well along its way, and it's been spun out into a startup company that's trying to commercialize it. And once you do that and go to the FDA, we can't change it anymore because we have a file at the FDA. So I'm not too much involved in it at that stage because I'm interested in developing new technologies and advancing this. So the company is trying to take this to the clinic and working on it now. But we had a lot of press on this. This is our little hip in a jar with my student, Allie Ross. The thing Allie was able to do was take this hip and insert a gene into those cells so that those cells could now make anti-inflammatory drugs right in your hip joint once it was transplanted. And she had it encoded where we just give a harmless drug that you swallow, and it would turn on the gene in your hip. And she showed that we could make just deliver exactly to your joint these anti-inflammatory drugs. Now, this got posted on Facebook, and my daughter was really mad at me because I got 4,000 likes. So that's where we are with that, but there are all sorts of things that can go wrong that we're foreseeing. So we want to cut those off at the pass. And one of the things that can go wrong is there's a reason your hip degenerated in the first place. There's a lot of inflammation in that joint. And inflammation is a natural response of the body to damage where it tries to bring in cells to fix what's going on, and normal inflammation is necessary for repair. But what happens is in this engineered cartilage, this red shows newly formed cartilage. If we expose that cartilage to inflammation, you can see it just wipes out our newly formed cartilage the way it wiped out our old cartilage, because a little bit of inflammation is good. Long-term inflammation we know is bad. It causes pain, degradation, and all of these other problems. And in fact, there's sort of this thought that we need what's called resolving inflammation. You need a little bit of inflammation, like if you cut your skin. You have an inflammatory response, but your skin heals. If that inflammation, and this is what resolving inflammation looks like. You first have the onset of inflammation, it resolves, and then later, it's called post-resolution, other cells come in to clean up the mess and get rid of the fibrous parts, get rid of any infection or anything like that. The problem is when you have chronic inflammation, and several diseases like these autoimmune diseases cause chronic inflammation where you have the onset, but it just doesn't go away. You get into this circulatory progressive inflammation, and it just stays on. And so diseases like rheumatoid arthritis or psoriatic arthritis and Crohn's disease, this is just unbridled inflammation where it just stays there. And there are drugs for this, but this is what they do. If you take anti-inflammatory biologics, they just totally wipe out your inflammatory response, and that's not good either. It doesn't look like this, where you have resolution. So our thought was, well, how do we get cells to turn this chronic inflammation into a resolving inflammation? And so what we wanted to do was reprogram those cells. Can we make those cells smart so they can sense the inflammation and don't get into this vicious cycle of continuously producing inflammation? So when I took biology in college and in high school, this is what we were taught a cell looks like, right? It's got a membrane and a nucleus and organelles. But as I mentioned to you, I'm an engineer. So I think of a cell more like this, where it's got integrated circuits and motors and sensors and so on. And as a bioengineer, we think of cells as a combination of the two of them. And we enter this field called synthetic biology, where we're trying to create cells that don't exist in nature, but they do what we want them to do. And I think of a cell now, a stem cell, not as a machine, but sort of like an iPhone. And we can reprogram that iPhone. We can insert DNA code and rewire it so it does what we want it to. And we can have it sense biological signals that we tell it to sense, and then program in genes that give it anti-inflammatory responses, block pain even, or turn on growth factors to enhance repair. And there's nothing better than a stem cell as sort of a blank slate, where it's sort of like a blank iPhone, and you can put in your own apps into it and reprogram it. So how do you program DNA and program a cell? Well, until a few years ago, it was really difficult to do. But a few years ago, this is our main target, which is these anti-inflammatory pathways that are used for autoimmune diseases. So things like TNF inhibitors, IL-1 inhibitors, IL-6 inhibitors. These are drugs that are given systemically at high doses for autoimmune diseases. And they're partially successful, but they have side effects, because once you block the immune system, you have a very high risk for infection, some potential risk for blood cancers, leukemias, but the disease just goes up and down. So we need something that can respond to that up and down instead of a constant drug delivery. So how do we reprogram the DNA? A few years ago, this technique was discovered called CRISPR, and you may have heard about CRISPR in the news. It's part of the bacterial innate immune system. And what bacteria can do is find a spot on the DNA that they recognize very precisely and cut it open. And a couple of researchers, Jennifer Doudna and George Church Feng Zhao at MIT, figured out that in a mammalian cell, you could take this bacterial gene and it could identify a very precise spot on the DNA. You could cut it open, and as you saw, I'll replay the movie, this protein goes, finds a spot on the DNA. It recognizes an exact code of 20 base pairs. When it recognizes it, it opens up and cleaves the DNA. And once you do that, you can now splice in the DNA that you want. So we make a second strand, and now we can cut and paste DNA. So we can remove whole sections. It's sort of like splicing an 8mm film. You can just put in exactly what you want, where you want it now, and we can rewire that cell. So once we decode the region that we understand in the cell and we want to do something with it, we can replace it with a new code. And so what we wanted to do was take that stem cell, which when it sees inflammation, it has a strong inflammatory response. So we went in and just cut out that inflammatory response. We deleted that gene and inserted in its place a drug, and the same biologic drug that you see on commercials called Enbrel. It's actually a genetic sequence for inhibitor of tumor necrosis factor alpha. It's the soluble receptor for it, so it binds to it and blocks it, but it's a natural product, so we can just insert the gene. So again, going back to engineering, it's like making a little feedback loop where we take a cell that would normally just produce an inflammatory response, and we tap into that response, give it a feedback loop so it shuts itself off. And what we did was create a cell now that whenever it sees inflammation, it creates the drug that blocks inflammation. And so it's self-regulating. We don't have too much drug, we don't have too little drug, and it only produces drug when it needs to. And you can see in cartilage, when it's exposed to this molecule TNF, it wipes out the red, which is cartilage stain, and these self-regulating cells produce the drug that inhibits it. This is the equivalent of Enbrel, the drug that you can buy for rheumatoid arthritis, and it protects the cartilage. So we now wanted to put this into bodies, and we thought the best way to keep something around for a long time is to turn it into cartilage because it doesn't need a blood supply. It lives for years. And so we took our scaffold, took our genetically engineered cells, grew a little piece of cartilage that's about the size of a nickel, about the same thickness, and this little implant now acts like we call them smart cells. So the cells don't do anything until they see inflammation, and you can see when they see inflammation, they make lots of drug, and when you take the inflammation away, they stop making the drug. So they're self-regulating now. So you don't have drugs pouring into your body, you only have them when you have a flare. And then we tested this in mice, and this is a strain of mouse called the KRN mouse. It gets rheumatoid arthritis. And so what we did was put those little implants, you can see under the skin of the mouse, they glow so we can see where they are, and they glow when they are activated. And what we found was that these little implants, the red is the ones that got the implants, they completely eliminated the pain in rheumatoid arthritis, and they reduced the swelling by 40% and the arthritis score by also about 40%. So we now have the system that only delivers rheumatoid arthritis drugs when you need them and completely eliminates the pain. It also eliminated all of the bone erosion that occurs with this. So we're very excited about this because we now have access and the ability to create cells that do what we want them to. We can tell them which sensors they have. We can add sensors, take sensors away. We can put drugs in that they need to make, and we can tune them to these different diseases. And think of all the diseases that we have that are a problem with our feedback system. So diabetes, we're not making enough insulin for the amount of glucose that we have. All of these autoimmune diseases, Crohn's disease, and so on, we have now a technique to try to address those. And when I go back to the iPhone analogy, we can make all sorts of different apps. So the ones that we've made so far are the Find My iPhone. So these cells light up so we can see exactly where they are when we put them in the body. The more drug they make, the brighter they shine so we can get a signal intensity. We can shut them off. Let's say you're going in for surgery and you need to stop making drug because they inhibit healing. We have this system where we can do what's called a tetracycline off system. So when you give them a little bit of tetracycline, it stops the gene expression. Let's say one of these cells gets away and it, for some reason, forms a tumor. We have a self-destruct button that we build into them. We give them a harmless drug, and you can just wipe them out if you need to as a safety switch. And then we're now making even more and more complex genetic circuits into them so they can really identify which inflammation pathway is going on, so we can actually form these little genetic circuits inside of them so they can respond to one drug or another drug or both drugs at the same time and really give you a very tailored, personalized drug response when you need it. And so we're taking that, and once you have this, your imagination can run wild. The latest area we've been working in is one of the biggest risk factors for arthritis is obesity. And years of studying this, we learned that it wasn't just being overweight, it's actually your diet also. We learned that different fatty acids in your diet can be very inflammatory. So you've probably heard omega-6 fatty acids, like in corn oil, can be pro-inflammatory. Saturated fats, like in animal meats and lard, are pro-inflammatory. And then omega-3 fatty acids, like fish oils, are anti-inflammatory. And we tested these and showed that, yes, in fact, these omega-3 fatty acids can prevent arthritis. They can actually induce regeneration of many of your tissues by shifting the balance of inflammation. So we thought, well, why don't we make a cell that does this for you? We wanted a cell that would basically take this meal and turn it into this meal. And we found an enzyme. This enzyme doesn't even exist in mammals. It's only found in worms and plants. But because it's the same DNA code, it works in mammals. So we put this gene, it's called FAT1, into our cells. And what FAT1 does is it converts omega-6 fatty acids into omega-3 fatty acids in your body. So basically, it converts your McDonald's meal into your IKEA meal. And we deliver this as a gene therapy now. We put this gene into a harmless virus that's been inactivated, so it can't multiply, but it goes and infects all of your fat and muscle cells. And now every time you have this fat in your body, it converts them into this gene. So now we've got this in mice, and what we found, this is mice that are on a regular diet. And if they have an injury, the black bar shows their arthritis score. They get a lot of arthritis if they have an injury. If they have this FAT1 gene, it reduces it. But if they're on a high-fat diet, it almost completely eliminates their arthritis caused by obesity and a joint injury. And here you can see the cartilage. Again, the red shows healthy cartilage. And when they get arthritis, you lose some of the red. The ones that have the gene therapy really preserve their cartilage. So this is probably the first thing of all of these that I'm going to jab myself with when it's ready with the FDA so I can enjoy my new Shake Shack down the street. So as you can see, we have all of these really exciting tools now that 5 years ago this wouldn't have been possible, and it wouldn't have been possible if I hadn't moved here. It's really the environment here. We have the #1 musculoskeletal research group and the #1 clinical group in the country, in the world. And that's really the reason I came here and to work with Dr. O'Keefe and the team that he's put together. With these tools, you can see now we can edit the genome of the cell, sort of the hardware of the cell. We can change the sequence. We can edit the epigenome, create these artificial cells. We call them designer cells because you get to design them, and then package them back as a virus or gene therapy, as a little implant that sits under your skin, making drugs for you for years at a time, or eventually as a whole joint replacement that will cover your hip and keep you from having a metal and plastic prosthesis until you really need it. And as I mentioned, when I started off, this was the future of engineering your body, was metal and plastic. And then 10 years later, it was engineering cells and tissues. And I think now it's a combination of the three of these: metal, plastic, cells, tissues, and engineering the genome, where we can read the code, we can edit the code, and we can make these cells do what we want them to. And this is just one of the labs. This is our lab in WashU Orthopedics, but we have, as you saw, 13 other faculty who are doing equally exciting and cutting-edge work. And I hope you hear from them at future sessions, but we've got people doing all sorts of really exciting work in bone and fracture repair, in the intervertebral disc, in the rotator cuff of the shoulder. Any orthopedic or musculoskeletal condition, we have one of the best people in the world working on it. And I didn't do any of this work. This was all done by a really phenomenal group of students and postdocs and staff in my laboratory, most of whom we recruited since coming here, and it's been wonderful having them come here. And none of this would be possible without support. And you can imagine how expensive these experiments are, particularly the ones where we do sequencing and gene editing. And all of these foundations and the NIH have really supported us over the years. So I will conclude there. And Birgit wanted me to show a video that my lab put together about this project called Smart and Soothy. My lab manager wrote a rap video, and they enacted it in the lab. So we're going to play that for you now. It's about cells that are smart, and they soothe your joints. Yeah, sure. So that's Chris. That's written and performed by him. So, well, I'd be happy to answer any questions.
Does CRISPR have a monopoly on that process that they use? I mean, or a patent?
Yes, so there's huge patent wars going on right now over the development of CRISPR. So the question, if anybody in here is whether there are patent issues with this technique. So two groups sort of simultaneously, one at MIT and one at UC Berkeley, fairly simultaneously invented it. And one filed it first, but the ones at MIT filed later and paid that extra fee to the patent office. So they were awarded it first, which is a great lesson. and so they have been in the courts for the last 3 years spending millions of dollars because this is worth billions of dollars. So they're fighting over it. So and it's constantly evolving. So the first generation was out of one type of bacteria. It turns out almost all types of bacteria have different forms of this protein, CRISPR, and Cas9, which is the CRISPR-associated protein. So there's different versions of it that people are now patenting and using that have different capabilities.
Doctor, there used to be a lot of political controversy over stem cells. Can you talk about it and where it is?
Oh, absolutely. So the question was about political controversy over stem cells. And the great majority, 99.99% of that controversy was over embryonic stem cells. And these are stem cells. Basically, if an embryo forms, it starts off as one cell that becomes fertilized, and then it forms every tissue in the body. So if you catch those stem cells early from a discarded embryo, you can use those to study development, and they have the ability to do that. So what we're talking about here are not embryonic stem cells at all. These are all either cells that we have right now, like in my love handles, or in any tissue in your body, or, as I mentioned, we can take skin cells and convert them into that embryonic state, but they're still just your own skin cells, things that they're not nearly as controversial. Probably the one good thing that's come out of all of these clinics that have popped up and started peddling what they're calling stem cells is the word stem cell is no longer so controversial because they are everywhere, even though they're not really stem cells that are being sold, but it's sort of taken that mystique away from and the confusion of what's an embryonic stem cell and why we might not want to use embryos to do this research. That said, that embryonic stem cell research is still very important so that we know what we're dealing with is actually truly a stem cell, and even understanding the effects that certain drugs or toxins have on embryos, we can test on a mass of cells that were created from embryonic stem cells versus, it'd be impossible to test them on embryos, obviously. So that there is a place for it, but the work that we do is all adult stem cells.
You had mentioned that as part of your research, private companies have been formed. Who benefits from those private companies?
Yes, so hopefully the patients, and that's really the main path to get something to the clinic is to form an entity that can commercialize it. And so the way those startup companies that form out of the university work is that the inventors of the patents get a fraction of the royalties, the institution gets a fraction when it's formed of the patent royalties, and then in the end, the companies go through so many dilutions by the time they become public that ends up being about 1% of the actual value once they're sold and they make it to market, from my experience with watching these things go through and having friends go through with it. So in the end, the venture capitalists benefit quite a bit. And if something really makes it to market, then everyone benefits. But there is ownership from the institution, the university, the hospital that support the research, and for the inventors.
Once you reprogram the stem cells, you can destroy it.
Once we reprogram the stem cells, can anything destroy?
Then you put it back into
Yes. So
destroy it.
yes, that's a great question. So can anything destroy the stem cells when we put them back in? And the problem is that once cells go back into your body, if they don't go to the right place, then they can't really form a home. And it's called a niche. The stem cell niche is sort of its home. So if it doesn't end up in that niche, it just ends up either degrading or getting attacked and disappearing. And the way we've overcome that is by encasing it in cartilage and making that little implant, that little nickel-sized implant, it protects it completely so other cells can't get to it and they can't crawl away. So that's one of the major issues that's sort of held back cell transplantation is we can inject a whole bunch of cells in the knee. We look a week later, they're all gone. There might be 1% of those cells left for the exact reason that you're saying. So that's been one of the biggest challenges that we had to overcome was to create that implant that stays there for months at a time.
When gets the RA implants will be FDA?
So when would these be FDA approved? So those are probably the furthest away. So the hip implant is really on its way, and if everything goes well, 2 to 3 years, it'll be hopefully first in humans. The RA implant, we've just been contacted by some of the big RA drug companies. The ones that make all of the current drugs, their patents go off in the next year or two, and they're looking for the next generation. We think even with everything involved in it, it's probably about a 10-year process to be able to get something, mostly because the FDA doesn't have a lot of experience with living cells or cells that have been genetically modified or combinations of cells and materials. So for this to be available in the US, it's probably about a decade of work, although in Europe or Asia, it could probably go a lot faster because these types of stem cells are in other clinical trials, not reprogrammed ones, but just stem cells are in good clinical trials in Japan, for example.
Will this be able to be used to address nerve degeneration?
Yes. So would this be able to be used for nerve degeneration was the question. And we think this can be used for any degenerative process as long as we understand what's causing the degeneration. So different forms of degeneration are caused by by different parts of your body or different chemicals attacking. So, for example, certain nerve degeneration issues are caused by what we call demyelination, cells that in an autoimmune manner attack the sheath of the nerve. So in that case, if we know what the target is, we can create cells that make the drugs that block that target. In other cases, like everyone asks about Alzheimer's or Parkinson's, until we really understand the process and we have a target, it's much harder to then create the drugs and the target. And one reason we worked on rheumatoid arthritis instead of osteoarthritis for these cells is we know the target for rheumatoid arthritis. It's tumor necrosis factor, TNF. That's where all the drugs are based on. For osteoarthritis, we don't have a great drug target yet.
Can you explain your association with Shriners Hospital? I see that in part of your title.
Sure, yeah.
And why that is important to you and how this all fits.
Oh, absolutely. So I'm a faculty member at Washington University in the Department of Orthopedics, but I also serve as the director of research for the Shriners Hospital. And the Shriners Hospital, for those, I'm sure you're all familiar with the Shriners Hospital, it's a pediatric hospital where the surgeons who operate there and the faculty are WashU staff and faculty. So I oversee the research program there. And, we're there, we are in the hospital, we're on the same floor as the children, and we see the kids. They're walking around. You can see exactly what you're trying to treat. So all my staff who are walking in, they see these kids in wheelchairs or with a halo or with leg braces on. And it's really an amazing experience versus being in a pure research lab where everyone around you is a scientist. Every day there when we walk in, we see the children that we're trying to develop cures for. So our research program there is really focused on the juvenile aspects, the children's aspects. So a kid who needs a hip replacement at age 13, we want to cure that. Or a kid who has juvenile arthritis, which is, the starts at 1 year, 2 years old, this sort of inflammatory disease with really no treatments for them, and they can't tolerate these massive drug doses. That's what it means for us to be there and why we target the juvenile forms of all these diseases in that laboratory.
Is there any reason to believe that this will actually be available from a cost perspective, covered by insurance, priced out of people's reality?
Yeah, so the question is about cost and insurance, and I think that's just absolutely essential for these types of therapies to get to people. They have to be covered by insurance. And that's really, based on the fact that they should be cost effective. By the time we're done with them, it should make sense for an insurance company to pay for these instead of paying for a joint replacement. And that's been really, in our startup company, that's sort of the business model, is say, all right, if you can delay a joint replacement by 5 or 10 years, you buy so much quality of life that it's worth doing this versus giving a child a joint replacement. But that's really front and center once you start thinking about commercializing versus being in a laboratory and doing the basic research that leads to this.
Take one more question. And Dr. Guilak and Dr. OKeefe will be available for questions later on after this.
Thank you.
Well, we have these leading edge presentations from time to time, and they're great. And sometimes they really make you think. Well, this one really makes me think. And we really thank Dr. OKeefe and Dr. Guilak for a very stimulating and innovative topic. And we know that there's some wonderful things going on down on the campus. I always see when we have these discussions, I feel lucky. And I feel lucky that we live in a town that has a resource like Barnes-Jewish Hospital and Washington University School of Medicine. The things that are going on there that affect our lives are just incredible. And my wife and I are very familiar with the orthopedic department at Washington University. We both in the last 5 years had our backs, for different reasons, redone. And not regenerated, but redone. And we're waiting for that. But it makes for a life-changing experience for us, and not everybody can get that in the country and have it in their own backyard, and we are just so fortunate to have that. I'd like to really thank the donors, the board members, and members of our staff at the foundation for coming here tonight. The importance of foundation support it goes without saying, and we really appreciate everything you do to support the foundation. It allows us to invest in innovative research and individuals to perform that research at Washington University and Barnes-Jewish Hospital, and come up with just life-altering solutions like this that you can hardly imagine. I also appreciate the fact that you put this on a level, and your slides put this on a level that I could understand. It was easy to follow, and I didn't get all of it, but I got a lot more than I thought I'd get, and it was really good. The other thing we'd welcome you to do is go look at the foundation website. It's got a lot of good things on there about the projects that we're doing all the time, and share it with your friends, or look us up on Facebook. So you'll see a lot of interesting topics. You'll see additional presentations like these, leading edge events that are coming up in the future, and we invite you to come and bring your friends, because we want to get the word out and really share what's going on at Washington University and Barnes-Jewish Hospital. So you can get a lot of good information there. Dr. Guilak and Dr. OKeefe will be here to answer any of your other questions if you'd like to meet them and discuss things with them. And I'd like to remind you that we have another leading edge event that's coming up in November. It's on a Tuesday, November 12th, and it's on leading edge nursing. And nurses are critical to our delivery of medicine everywhere. And we've got a great event coming up with some of the top nursing professors at the Goldfarb School of Nursing. So put that on your agenda if you can. And thank you for coming, and thank you to the doctors for a very stimulating discussion.